Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221, and this is lecture number 10 of the series of our online lectures. Today the topic for lecture is genetic code. The outline for today's lecture is we'll talk about introduction, then we'll talk about genetic code, we'll talk about genetic code table, then characteristics of genetic code. We will talk about nonsense codons, Hoobling phenomenon will be discussed. So this is the outline for today's lecture. Now we move to our lecture. Now the flow of genetic information, this is just to recall you, according to the central dogma of molecular biology. We have DNA. The information from DNA are transcribed to RNA. DNA itself, from one generation to another, or from one when the cells are divided, it is passes through the process of replication, in which it is its copy are produced. In another way, when the information is in DNA is transcribed to RNA, and then this RNA is translated to proteins. So the central dogma of molecular biology describes the two-step process, transcription and translation, by which the information in genes flow into proteins, that is from DNA to RNA to protein. So this slide tells us about the central dogma of molecular biology. Now this messenger RNA, obviously it consists of nucleotides U, G, C, G, C, U, A, G. This is how it is showing. So this is the messenger RNA. Now these are the different amino acids: methionine, serine, leucine, lysine, and arginine. Here is the codon. We will talk about this in detail how it functions. This is the transfer RNA. Here again, the transfer RNA that pick the protein according to the code that is provided by the messenger RNA transcript. So the pathway of protein synthesis is called translation. We will talk about translation in detail in the coming lectures, because the language of nucleotide sequence on messenger RNA is translated into the language of an amino acid sequence. The process of translation requires a genetic code through which the information contained in nucleic acid sequence is expressed to produce a specific sequence of amino acid. Now the letters A, G, T and C correspond to the nucleotides found in DNA. They are organized into codons. The collection of codons is called genetic code. So this is what is genetic code. Collection of codons is called genetic code. Now we have 20 amino acids, and for 20 amino acids there should be 20 codons. Each codon should have 3 nucleotides to impart specificity to each of the amino acid. For a specific codon, nucleotide 4 combinations, nucleotides 16 combinations. If it consists of, say, if it consists of single, then there are 4 possibilities, or 4 combinations possible, which are A, G, T and C. In case there are 2, there are 16 combinations possible. In case it consists of 3 nucleotide, there are 64 combinations possible, most suited for 20 amino acids, because neither 4 nor 16 can code for all the 20 amino acids. So we have 63 nucleotides that code for 64 combination, and it is most suited for 20 amino acids. So the genetic code, that is DNA. Now this is the RNA with 3 bases code for one amino acid. So it can be like ATG, it can be CTA, it can be GGC. So these different three bases, they code for one amino acid, here also and here also. So these, for example, in this situation we have these three codons. They can code for three 9 codons that can code for 3 different amino acid, combination of 3 codons for 1 amino acid. Now, genetic code is a dictionary that corresponds with sequence of nucleotides and sequence of amino acids. Words in dictionary are in the form of codons. Each codon is a triplet of nucleotides, 64 codons in total, and 3 out of these are nonsense codons, 61 codons for 20 amino acids. This is the table. So like we have, we can possibly have triple U, UUC, UUA, UUG. So these two, they code for this amino acid, these can code for this leucine, these 4 can possibly code for serine, these 2 for tyrosine, UAA and UAG, they are stop codon along with UGA, UGU, UGC for cysteine, UGG for tryptophan. Similarly, now in this table we have all the 64 codons that are possible. Among these 64, these 3, UAA, UAG and UGA, they are stop codons, and the rest of the possible codes are shown for different amino acids that we have. So genetic code have certain characteristics. One of the property, or one of the characteristic, is specificity. Genetic code is specific unambiguously. A specific codon always codes for the same amino acid. For example, triple U codes for phenylalanine. It cannot code for any other amino acid. So here you can see this, or for example CCU or CCC, it codes for proline. So it cannot code for any, for example, it cannot code for leucine. It codes only for proline. In this case, these codons are for threonine, so they only code for threonine. They cannot code for, say, methionine or valine or adenine. So it cannot code for any other amino acid. Now, genetic code is universal. Universal in all living organisms. Genetic code is the same. The exception to universality is found in mitochondrial codon, where AUA codes for methionine and UGA for tryptophan instead of isoleucine and termination codon respectively of cytoplasmic protein synthesizing machinery. So these are some of the exceptions in this universality of genetic code. This has to be kept in mind. AGA and AGG code for arginine in cytoplasm, but in mitochondria they are termination codons. So these exceptions has to be kept in mind. Now, genetic code is redundant. What it means? Genetic code is redundant. Genetic code is redundant, also called degenerate. Although each codon corresponds to a single amino acid, but a single amino acid can have multiple codons. Except tryptophan and methionine, each amino acid has multiple codons. So you can see here these 2 codons, they can code for phenylalanine, these 2 can code for leucine, these can code for isoleucine, this can code for methionine. You see, we talked about methionine and tryptophan. These are the 2 which are only coded by a single codon, here UGG in case of tryptophan, and in case of methionine we have only AUG. Rest of the amino acid can be coded by multiple codon, or can be coded by different codons. In case of threonine, you can see these 4 different codons can code for threonine. Alanine, you can see these 4. Phenylalanine, you can see these 2. Leucine, you can see this these 2. So there is the property of this redundant. Genetic code is redundant, and that is called degenerate. So except for tryptophan and methionine, where each amino acid has multiple codons. So this has to be kept in mind. Remember that I talked in the introduction that there are 64 different codons possible. So these 64 different codons, they code for 20 amino acids. Among these 64, there are 3 which are stop codons: UAA, UAG, UGA, and the rest of the 61 different codons, they code for the 20 amino acids that we have. Now, genetic code is non-overlapping and non-punctuated. It means all codons are independent sets of 3 bases. There is no overlapping. Codon is read from a fixed starting point as a continuous sequence of bases taken 3 at a time. The starting point is extremely important, and this is called reading frame. There is nonsense codons. There are 3 codons out of 64 in genetic code which do not encode for any amino acid. These are called termination codons, or stop codons, or nonsense codons. The stop codons are UAA, UAG, and UGA. They encode no amino acid. The ribosome pauses and falls off the messenger RNA while one set encounters the stop codon. Here you can see here the UAA, UAG, and UGA. They are stop codons. So these stop codons, they don't code for any amino acid. It results in the pausing of the ribosome and falls of the messenger RNA. Initiator codon is very important. AUG is the initiator codon in majority of proteins. In a few cases, GUG may be the initiator codon. Methionine is the only amino acid specified by just one codon, AUG. Now, there is a phenomenon associated. It's called Oobling phenomenon. The rules of base pairing are relaxed at the 3rd position so that a base can pair with more than one complementary base. Some transfer RNA anticodons have adenosine at the 3rd position. Adenosine can pair with U, C, or A. This means that we don't need 61 different transfer RNA molecules. Only half as many are required. So in case this Oobling phenomenon was not there, we would require 61 different transfer RNA. But because of this Oobling phenomenon, some transfer RNA anticodons have this adenosine at the 3rd position. It can pair with U, C, or A, which results in a situation where we don't need 61 different transfer RNA molecules. Only half as many are required. Now, Oobling phenomenon. First 2 bases in codon in messenger RNA in 5' to 3' base pair traditionally with the 2nd and 3rd base of the anticodon in transfer RNA, 5' to 3'. Non-traditional base pairing is observed between the third base of the codon and the first base of entry codon. The reduced specificity between the third base of the codon and the complementary nucleotide in anticodon is responsible for ubbling. So, this reduced specificity between the third base of codon and the complementary nucleotide in anticodon is responsible for ubbling. Now, ubbling phenomenon, here this transfer RNA first base and messenger RNA third base, here CG, this base pair is traditional. In case transfer RNA first base is A and messenger RNA third base is U, then again the pairing is traditional. In case transfer RNA first base is U and messenger RNA third base is A, then the base pairing is traditional. Now, in case the transfer RNA first base is U and the messenger RNA third base is G, in this situation, traditional pairing is not possible. This results in non-traditional base pairing. Now, in case of G in the first base of transfer RNA and C in the third base of messenger RNA, again the pairing is traditional. But in case of first base of transfer RNA is G and the third base of transfer messenger RNA is U, the base pairing is non-traditional. In case of in other situations where the first base is I in the three cases and the third base is U, C or A, in this situation the ubbling, because of the ubbling, the base pairing is again non-traditional. So there are different possibilities that can either be result in the base pairing in traditional way or in non-traditional way. So this thing has to be kept in mind while studying genetic code. Now, ubbling phenomenon, proline has 4 codons CCU, CCC, CCA, CCG. The first 3 codons can be recognized by a single transfer RNA having an sign at the first place, IgG 5' to 3'. So you see here, because of this ubbling phenomenon, although proline has 4 codons, the first 3 codons, they can be recognized by a single transfer RNA. So in this situation, for example, proline does not require 4 different transfer RNA because of the ubbling phenomenon. It can the first 3 codons, they can be recognized by a single transfer RNA having an sign at the first place, IgG 5' to 3'. Now, it has clinical significance. Mutation can be well explained using the genetic code. Point mutations that can be silent, can be missense, can be nonsense, can be frame shift mutations. So silent mutations, single nucleotide changes A to G, same amino acid is incorporated, mutation goes unnoticed. So now in this situation you see ATG code for methionine and GAA code for glutamate, CCA for adenines, CGT code for glycine, ATG code for methionine. In this case, you see GAG, this is a mutation, but still it codes for the same amino acid. In case of BCA, this is the report for L and for glycine. So in this situation, this is a silent mutation, although there is a mutation, but it goes unnoticed. Mutation goes unnoticed. Missense mutation, single nucleotide change A to C, different amino acid incorporated, loss of functional capacity of protein. So in this situation, for example, this code for histidine, replacement of a single nucleotide results in proline, incorrect amino acid, which may produce a malfunctioning protein. So these are the DNA bases. These are the different codons for different amino acids. You see single base pair, a single nucleotide change here results from histidine to proline. So this is incorrect amino acid, which can possibly result in malfunctioning of protein. There is another situation where it can result in nonsense mutation. Single nucleotide change from C to T, stop codon is generated in messenger RNA, represented by UAG. Premature termination of chain may be incompatible with life. So here, in this case, there is a change, a replacement of a single nucleotide to T results in incorrect sequence, causes shortening of protein. So this is an example of nonsense mutation. Now there is another situation which is called frame shift mutation. Insertion or removal of a basis can alter the reading frame with resultant incorporation of different amino acids. So you see here frame shift can result in, now the reading frame is changed. It can result in insertion or removal of bases, can alter the reading frame with resulting incorporation of different amino acids. So okay, instead of methionine glutamate alanine glycine, now methionine glycine histidine. So this is a frame shift mutation. Now this in this slide we summarize the lecture. It says that genetic code is unambiguous, universal, degenerate, commonless, and non-overlapping. UAG, UGA, and UAA are termination codons. AUG is the start codon. Mutations can be well described using genetic code. So this was all about genetic code. I hope you enjoyed the lecture. If you have any questions, please feel free to ask me. I'll be happy to answer. Thanks for watching.